Hi, I'm Byron Caughey out here in the wide open spaces of Montana at the Rocky Mountain Labs, which is part of NIH, or the National Institutes of Health. I have no idea about making selfie videos, but here goes anyway. First, I want to thank Jenny Cole, Kathy Esposito, Rhonda Fansler, Janine Cook, and Linda Parrish for being so generous in funding our efforts to test molecules called antisense oligonucleotides, or ASOs, as potential drugs for CJD. ASOs are drugs that can block the production of specific proteins in the body and in the brain. Since prion diseases are due to the accumulation of toxic forms of a specific protein called PRP, as you know, our basic idea is to shut off PRP production in the brain using a specifically designed ASO. Now, Stan Prusner's lab, along with a company now called Ionis Pharmaceuticals, had tried this approach a few years ago and had some initial signs that this approach might work. Since then, the people at Ionis have learned better strategies for designing and delivering ASOs to treat neurodegenerative diseases while minimizing side effects. Indeed, recently, Ionis has had some major successes in developing ASOs to treat other diseases, and one of those compounds was recently approved by the FDA to treat spinal muscular atrophy. Sonia Valop and Eric Minical, whom you may know already as passionate and really brilliant students at the Broad Institute, had surveyed the field and decided that ASOs were currently among the most promising of therapeutic approaches for prion diseases. They asked for our help in revisiting this approach and coordinated this multi-center project. Holly Kordaziewicz and Ian Zhao at Ionis oversaw the production and screening of a huge number of new ASO molecules that, based on their pioneering understanding of how ASOs can work, were designed to turn off the production of PRP. They sent us a number of the most promising ASOs for testing in prion-infected cell cultures and in animals. We needed your support to do these expensive experiments because, as you may know, funding for prion research, and in fact scientific research in general, for that matter, has fallen flat in recent years. We were excited to find a couple of new ASOs from Ionis that were highly effective at reducing the formation of the abnormal form of prion protein that causes prion diseases such as CJD. Lynn Raymond first saw evidence of this in prion-infected cell cultures. Greg Raymond, Brent Race, Katie Williams then tried two types of treatments in rodents, each involving the microinjection of ASOs into the brain. In humans, these injections would be done at the base of the spine. One of our experiments included an ASO pretreatment prior to the inoculation of prions into the brain. After that, repeat injections of the ASO were given every 60 days. This experiment was meant to approximate the situation that people who carry a prion disease-linked mutations might find themselves in. That is, the patients know they're at risk before they're actually sick and might then be able to start treatment before much damage is done in their brain. We were really happy to see that in this experiment, the ASO-treated animals lived 70 to 90% longer than the untreated animals. A second and more challenging strategy was to infect the animals first with prions and wait until they were within a few days of showing clinical signs of disease, that's about 120 days, before actually treating them with ASOs. At that point, they already have a rip-snorting prion infection in the brain. This scenario would be closer to the common situation faced by patients who are having the first signs of disease before they even seek therapy. These studies are still in progress, but are looking quite encouraging. And we hope to be able to give you a more complete report on the results soon. Until then, I want to thank you again for making all of this possible and wish you all the best in facing the huge challenges presented by these diseases to patients and their families. Thank you.